Hi, welcome to this video series. My name is Christina Kuiper. I hold a PhD in biology, but I'm a data scientist by heart and I work with it for more than 10 years by now. I have a special focus on AI tools in biology, so I'm really happy that you chose this course. Basically, you chose me to teach you how to use AI tools in biology and specifically AlphaFold 3. Importantly, all the tools that we will use in this course are free to access and require no programming skills, making them accessible to all researchers. In this video, we will explore what is AlphaFold 3, we will look at its pros and cons, you will learn how people are actually using it and who is allowed to use AlphaFold 3 because not everybody is allowed to do that. And of course, I will also tell you what makes AlphaFold 3 so powerful.
Let's start over with the introduction to AlphaFold 3. Molecules are so important for understanding our life. Molecules can make up proteins, these tiny machines that fulfill a multitude of different functions. In many cases, we still don't understand these functions in detail. But this would be so important to understand many different diseases and to find solutions for the patients. Proteins often play a major role in these knowledge gaps, and so all researchers in these fields were extremely happy when AlphaFold opened its doors, as it really has the potential to help solving many of these problems in life sciences. You may wonder why, and I will give you the answer throughout this video. But let me say one thing in advance. It's also because of AlphaFold's extreme precision and the detailed metrics that allow the researcher directly to interpret if the structure was predicted correctly or not. AlphaFold 3 was developed by Google DeepMind and Isomorphic Labs and builds up on AlphaFold 2, but with a massively improved architecture. If you are interested in all the detailed upgrades that AlphaFold 3 has in comparison to AlphaFold 2, I can only encourage you to read this publication. AlphaFold 3 has a lot of advancements, out of which the ability to predict complexes with other molecules than just proteins is probably the most crucial one. And you know what? This is possible because the developers changed what is being recognized as a unit by their deep learning framework. In AlphaFold 2, these units were amino acids, and so AlphaFold 2 could predict protein structures. Instead, in AlphaFold 3, they changed the unit to being any molecule, and this unit can be an amino acid, but it can be also an ion, or for example adenine as part of a DNA strand. It can be a ligand, or a small post-translational modification, or other biologically relevant small molecules. Let's have a look at the pros and cons of AlphaFold 3. Despite its high accuracy, which we have talked about already, AlphaFold 3 comes also with some negative sides. First of all, dynamic structures cannot be predicted accurately, as the molecules the model was trained on are from the PDB and they are static as well. But in reality, molecules undergo conformation changes all the time and can be highly flexible, which is not possible to address using AlphaFold 3. Not yet, at least. Also, RNA-specific interaction remain difficult to predict, especially when it comes to the typical complex tertiary interactions. Well, some researchers therefore criticize that it's still a protein-centric architecture. Fair enough. And the prediction is always as good as the training data only. For example, if you are interested in a rather rare plant protein that does not have solid evolutionary data yet, you may see your prediction ending up with a rather low score. And the last con. When it comes to powerful tools, as AlphaFold 3 is, I believe, there are always some ethical considerations. In this context, they go towards bioengineering and custom protein design with the potential for misusages. Don't worry, there are also more pros. First of all, it's easy accessibility. This is not only the freely available code on GitHub, which I will place the link to in the description below, but it's also the existence of the web interface that allows non-coders to access it and try protein interactions by themselves. You have a huge advantage if you use AlphaFold 3 because it reduces experimental time, money and resources, which makes it a tool that can be sustainable. And it should be used by anyone in mechanistic research if a question can be answered by a prediction. You just need to make sure that you do your experimental validations if one of the predictions was meaningful for your research. And most importantly, AlphaFold 3 contributes to urgent research needs, such as drug discovery and understanding infectious diseases, so it has an important global impact. Let's talk about who is allowed to use it. The AlphaFold server, as well as the code which Google DeepMind has placed on GitHub, is meant for non-commercial use only. And if you want to publish a prediction made here, you should cite their paper. It is not allowed to use the output for dogging or screening tools or to train more machine learning models, of course. Let's come to a really important question, which is big. How does it help researchers actually? To answer this question, I brought some examples for you. Let's start with the first one. Many scientists, including myself, use AlphaFold 3 for protein-protein predictions and protein-ligand predictions. Also, for example, Tian et al. Used AlphaFold 3 in dynamic simulations to identify zinc ion binding sites in caspase 3, which is an enzyme essential for apoptosis. It's the programmed cell death. As a second example, some people use it to predict large protein complexes or assemblies. And there's another example for this. Anyana et al. Modeled the assembly of a viral enzyme, which is the SARS-CoV-2 main protease Mpro. It was not only about its dimerization, but also about higher order states, which relate directly to understanding viral replication mechanisms and thereby drug targeting. Very important. You can also use AlphaFold 3 in combination with genetics. If you want, for example, a prediction of a structural change based on a mutation. This, for example, Yao et al. Did for improving our understanding of the Alzheimer's disease. They wanted to understand how mutations affect the misfolding and the aggregation of the beta protein, which is a hallmark of the pathology of Alzheimer's disease. And one last example. AlphaFold 3 is instrumental for nanobody antigen or antibody-antigen predictions. For example, Eshak and the colleague used a set of 70 nanobody antigen complexes to rank the AlphaFold 3 ability for its prediction accuracy of nanobody epitopes. AlphaFold 3 could outpace AlphaFold 2 here by more than 10%, and of course the authors also listed the pitfalls for the specific use case, which is maybe important for some companies who have a license agreement with Google DeepMind. Feel free to check out the publications to these examples so you get a better understanding of how you can apply AlphaFold 3 also for your research. With all these important aspects, I think you got that AlphaFold 3 is a powerful tool. But this innovation not only improves our understanding of biology, it also opens up new avenues for scientific exploration and therapeutic development. By providing highly accurate predictions of protein structures, AlphaFold 3 serves as a powerful tool for researchers across various disciplines, including molecular biology, biochemistry, biotechnology, drug discovery, and so on. It allows scientists to accelerate their research processes, as they can now rely on computational predictions to a certain extent, instead of solely on time-consuming experimental methods. And this capability is particularly vital in addressing urgent global challenges, such as developing vaccines or understanding disease mechanisms at a molecular level. Let's have a look at why is AlphaFold 3 so powerful. It's a very strong tool and a must-have for your scientific toolbox because the model is trained on a huge amount of data, using an advanced neural network. There are multiple aspects belonging to that. First, it uses protein structures from the Protein Data Bank, short PDB, that were released before April 2018 as a ground truth training data. And this includes over 170,000 experimentally determined protein structures. Moreover, the training also incorporates multiple sequence alignments from a very large protein sequencing database that included hundreds of millions of protein sequences which were clustered into families to learn evolutionary relationships. Moreover, AlphaFold 3 training used advanced techniques like cross-distillation with the structures predicted by previous AlphaFold versions to improve predictions on disordered regions. And that apparently really worked. The training process itself needed massive computing power running on over 100 to 200 GPUs over an extended period of time. Then the AlphaFold protein structure database now contains predictions for over 200 million proteins from over 1 million species. That's a number, huh? But this reflects how broadly the training data represents the known protein universe. All of this gives it great performance, high accuracy, and through the AlphaFold server web interface also wide accessibility. This combination and especially the precision was not possible before, making AlphaFold 3 a breakthrough tool in life sciences. Let's sum up. AlphaFold 3 represents a monumental step forward in our understanding of proteomics, and it influences many fields in life sciences. It has the potential to revolutionize a multitude of scientific fields. I believe that anyone interested can use it to produce meaningful results with the methods that I will teach you later on in this series.
That's it already for today. I hope you learned something. In the next video, we will explore the AlphaFold server web interface. And I will explain you the metrics necessary to predict your protein of interest or your structure of interest with a high confidence. This video is part of a full workshop on AlphaFold 3 as part of a series of AI tools in biology. You can go to my website if you're interested and buy it. I would be happy. And then, thanks for watching this video. If you found it helpful, please feel free to subscribe and stay tuned for more content on AI tools in biology. And if you have questions or comments, please feel free to put them below. I'm here to read them and I will be happy to help with your questions.